Okay. So yeah, like I was saying, and I know not everybody's here yet, but this is recording, so this will actually let me say that last. Uh, what are some good updates that I can give while we're recording before everybody gets here? Okay, so for the exam, it is graded. Everything's up there. The only thing that's not accounted for is or are the extra credit points because the people who usually get the extra credit points didn't, which means I think I made a mistake on which word is the correct word. Um, and the people who usually get those extra credit points, they all answered the same wrong question. So I assume you guys watched the video and you got the correct answer, and I just have it down there marked as wrong. So I need to figure out what the correct answer is, and then once that happens, then I'll go back in and make adjustments if needed. Also about the exam, like I said in the announcement, now that we've done exam 3, we can no longer redo exam 2 or obviously exam 1. That's been like that for a while. But if you guys meet with me, like office hours or any other time that we can arrange it, if you meet with me online and we go over your exams and talk about the questions you missed and talk about why they're wrong and why the correct answer is the correct answer, all that good stuff, then you can get a 5% boost to your exam grades. So that would be very helpful. Plus, keep in mind, your final exam is based on the final exam is written using the first 3 exams. So those first 3 exams are still useful. Those are still your study guides. But if you're going to study them, it would help to have all the correct answers. And to get all the correct answers, you need to meet me online and get them. So yeah, that's that for the exams. Um, Start sometime today or tomorrow. I'm going to re-release exam 3 for you guys to retake it. I know a lot of you haven't um, for the first 2 exams, but I highly recommend it. Like I said, at the very least, it'll give you practice for the final. Um, yeah, so if you retake the second exam or third exam, as usual, it'll be your first grade and your last grade averaged together to give you your new grade. Unless your new grade is lower, in which case we'll just keep your original higher grade. So again, there's nothing to lose. And when you retake the exam, it's not timed, it's open book. So there's nothing to lose. I don't know why more people don't do it. At the very least, even if you've got a good grade on the exam, again, it's just good practice to help you prepare for the final. So, any questions about exams in general? All right. Speaking of exams, one last thing. Since we just finished exam 3, obviously everything we do moving forward, including the chapter we've already started, chapter 13, that is going to be on the final. So chapter 13, chapter 20, and chapter 21 are going to make up half of the final. So about 50 points of the final will be made up of these last 3 chapters, and about 50 points and 50 questions on the final will be made up from the first 3 exams. So I'm going to take 50 questions randomly from the first 3 exams to write your final. So any questions about exams? All right, before we get started, I'll give you a quick second to get ready for this. But since we're all meeting online, we're just going to do attendance words again. Sadly, they won't be for extra credit, but be prepared. So if you need to grab something to write with, go ahead and grab something to write with. I'm going to go through them quickly, mostly just because we're in a rush because of not having internet service on Monday and not being able to give a lecture. So we're behind today. So I'm about to jump into this. I'm going to jump through it quickly. Please don't be hesitant to tell me to slow down or ask me to hold on or anything like that, because I am going to go quickly. We are recording. In the very least, if you don't feel comfortable asking me to slow down or asking me to repeat myself on this video, or excuse me, on this live stream, then just meet me afterwards if you want, during office hours, and say, hey, you're really flying through this. Can you please repeat this again? So anybody have any questions about anything before we get started? All right. Let's jump back into chapter 13. Okay. We were talking about mutations because, as we know, and the most important thing for the exam anyway, is that mutations are where, you can't have natural selection without variation, and you can't have that variation without mutations, right? So without mutations, there is no variation, there is no natural selection. And I'm pretty sure we left off on this slide right here. Talked about how sometimes a mutation can be bad, like using sickle cells as a disease. Generally, it's a bad thing, but sometimes that can also be good. Again, using sickle cells as a disease or as an example, because um, if you have sickle cell, I think you're either completely, you definitely won't get malaria, or you're at least very less likely to. So anyway, moving forward, chromosomal mutations that delete, disrupt, or rearrange many gene loci at once are almost certain to be harmful. So basically, that's your book's fancy way of saying the bigger the mutation, the more likely it is to be harmful. But duplications of a gene or small pieces of DNA through errors in meiosis. Oops, excuse me. Sorry. They can provide important sources of genetic variation, which again, without that, without genetic variation, we don't have natural selection or any evolution, and that eventually leads to new genes with novel functions. So again, there are new genes, obviously, because we have evolution. There are genes that exist now that haven't always existed, and these all basically come from mutations, and that's what this slide is basically trying to say. So, any questions about this slide? And the first word for attendance is this right here. All right, so no questions about this slide? I'm going to talk about this really quickly because I'm not going to test you on it, but I should point out that everything we basically this discussion is usually about eukaryotes, which is what we are, right? We're the more complicated organisms. But in prokaryotes, mutations quickly generate genetic variation in a population. And I'm going to explain why, even though you don't need to know for the exam. That's because bacteria are haploid. See, they have this one chromosome, right? We're diploid. We have one of each chromosome. So if we have a mutation on one of our chromosomes, then that's okay. And then you go through meiosis, right? So, half of those chromosomes will be in your sperm or egg, and the other half, the mutated one, will be. So that's unlike with bacteria, because with bacteria, 100% of the alleles get it. That's all we're saying there. And again, I'm not going to ask you any questions about that on the exam. I'm also not going to ask you this, but it's interesting information to know. On average, animals and plants have about one mutation for every 100,000 genes per generation. So for independent work, you could look that up. Like, how do we know that? And also, that's a pretty broad number, and it does say on average, and it does say about. So even though I'm not going to ask any questions about that on the exam, it is worth diving into, I think, if you're interested in it for independent work. Another thing about animals and plants, unlike bacteria, we have a relatively long time span between our generations, right? So we think about like a bacterial infection that happens quick, right? Because they reproduce quickly because they're single-celled organisms. So it's a very quick turnaround, unlike us, right? So if one of you sitting here right now watching this has a mutation in the right spot, mind you, it has to be in your ovaries or your testes or somewhere down there, right? If you happen to have a mutation, and then you happen to pass it along, right? It doesn't happen until you reproduce with another human, right? And even then, if you only reproduce once, the chances of you passing down that mutation are 1 in 2, right? So again, having a long time span reduces the number of mutations, or at least the number of transmissions and mutations. And like I already said in the previous slide, when we were talking about how prokaryotes are haploid, I pointed out that humans and other animals and plants have diploid genomes that prevent most mutations from significantly affecting the genetic variation from one generation to the next. Because again, if you have a mutation on chromosome number 2, just throwing out that one as an example. If you have a mutation on the chromosome number 2 that you got from your mom, that when you produce your gametes, your sperm or your egg, it's only a 50% chance you're going to pass along that mutation for chromosome number 2, because your kid might get the chromosome number 2 that you got from your dad that's not mutated. So any questions about this slide, which for the exam is not important? Okay. You guys are acting like it's a Monday. It feels like it. So let's talk about sexual reproduction really quickly, because we already kind of have been talking about it. So let's officially talk about it. Sexually reproducing organisms, or when it comes to sexually reproducing organisms, most genetic variation in the population arises from a unique combination of alleles that the individual inherited. So it's not necessarily going to be a test question. Basically, what we're saying is, again, when we're talking about sexually reproducing organisms like eukaryotes as opposed to bacteria, basically what this is saying is it's just more complicated, right? Um, because again, not only are we dealing with mutations and the fact that we're diploid, but we also have these three random components, as you already know. And this is stuff you needed to know from the last exam. And here it is coming back again, right? Independent assortment, right? So when all those chromosomes line up, those homologous chromosomes line up in metaphase of, or metaphase one, right? Metaphase one of meiosis. It's not always like mom on one side, dad on the other, right? The way they line up is independent of each other. And we also learned about crossing over in chapter 8. And remember, that's when these chromosomes are next to each other. Those homologous chromosomes are next to each other, and they swap chunks, right? So you might have a mom chromosome with a small chunk of dad on it, and a dad chromosome with a small chunk of mom on it. And of course, the idea of random fertilization. So basically, all these things, as you learned in chapter 8, where we talked about, hey, where does all this genetic variation come from? Well, now we're just repeating it again because that genetic variation is a very important component when it comes to evolution. All right, so any questions about this slide? Okay, let's talk about populations as the units of evolution. This is very important. People are going to miss this. I'm telling you now, someone's going to miss it, and it's going to make me sad. Just like I said, someone's going to miss the question DNA to RNA to protein, and it's going to make me sad. And I'm going to make it worth 5 points, and that's what I did on exam 3. And someone missed it, and I was sad. Anyway, populations is the unit of evolution. Before I even jump into this bullet point, what you need to know for the exam, like I've already said, is individuals do not evolve, right? Only populations evolve. Yes, individuals change and they adapt, but they don't actually evolve. The definition of evolution is it's a change in a population. So let's talk about it. The evolutionary impact of natural selection is only apparent in the changes in a population over time. Here's the first time I've officially given you the definition of a population. You need to know it. We're going to talk about it in other chapters. You need to know what a population is. I'm not sure if I'm going to ask you, but I'm definitely going to be using this term. I'm going to be using it with terms that are similar, and you need to know the difference between the terms. So for now, let's make sure you understand what a population is. It's a group of individuals of the same species that live in the same area and are breeding with each other. All right. So, think about the WVSU's campus. All the squirrels and all the cats on the campus are not a population because they're not the same species. And obviously they're not breeding with each other. Then again, if you think of all the gray squirrels in North America, right? They're not all necessarily the same population because the gray squirrels on WVSU's campus are not in the same area and therefore are not breeding with the gray squirrels that live in North Georgia, right? So again, a population, same species, they're living in the same place at the same time. They're breeding with each other. They're affected by the same conditions, like if there's a food shortage or a storm or whatever, they're all dealing with the same stuff. And that's what a population is. Any questions about what a population is? The next word for attendance is over. Different populations of the same species may be geographically isolated from each other. I read through this quickly because I accidentally jumped the gun and described it already, talking about the different places of the squirrels. But because of that, the exchange of genetic material over never or rarely occurs. So again, it's very rare that you're going to find the squirrel from New York mating with a squirrel from Alabama, for example, right? That's possible. One might accidentally land on a train and get taken all the way, across the country and then jump off and mate. Who knows? But your book also points out that such isolation is common with populations that are confined to different lakes or islands. So yes, again, gray squirrels, sure, it happens, but not often. But it's even less likely to happen when populations are on lakes or islands. Not that I'm going to ask you about that on the exam, but let me show you a picture and give you an example. See, so imagine, the fish population, whatever species, I don't know. I'm not really good with freshwater fish. Let's say there's a smallmouth bass in each one of these little ponds, right? The smallmouth bass, let's just call it bass because it's easier to say. The bass in this little pond are probably not going to breed with the bass in this pond or the bass in this pond, so on and so forth, right? Because it's not like they're just going to get up and walk over. However, I'm not saying it never happens because think if there's maybe a big flood, then, this might become flooded with water. And yes, the fish might be able to swim from one pond to the other occasionally. And then the water levels go back down. And then it doesn't happen as much. Or even who knows, maybe like an eagle swoops in, grabs a fish, tries to fly over there, and then accidentally drops it and it lands in there. And then there you go. But the point is, um, when populations are isolated, they're not interbreeding, they're not exchanging genetic information much. It's very rare, but it does happen. So, any questions about that? Another thing I want to point out that your book doesn't necessarily point out, but this is a good picture to talk about it. Imagine if the water wasn't always like this. Imagine if the water was, I don't know, 10,000 years ago, the water level was 20 feet higher, in which case this would all be one gigantic lake, right? So imagine that you've got that one population of bass. They're all pretty much similar because they're the same population, right? But then the water levels recede, and now you've got the population A in this lake, population B in this lake, population C in this lake, whatever. Eventually, they might start looking different, right? Because Lake A might be really deep, and Lake B might be really shallow, and Lake C, I don't know, maybe because of the way it's open without trees, maybe it's getting more predators coming in. I don't know, whatever the case is, all these fish are living in different conditions. So over a long time, even though they all started off as the same species, eventually it's very likely they would become very different. So much to the point where they might actually become eventually different species. But that's a whole different discussion that we will have later. I just wanted to go ahead and preview that now because this picture was a good picture for it. So any questions about this? Okay. At the population level, biologists focus on gene pool, which consists in this. You're already familiar with gene pools because this is what we've been doing in lab. The gene pool consists of all copies of every type of allele at every locus and all the members of the population. So, right, basically, a gene pool is all the genes and all the alleles, right? All the different versions of the alleles in every member of the population. That's what a gene pool is. Oh, sorry. Any questions about what a gene pool is? All right. Now I'm going to go through this again pretty quickly because we've discussed this in lab. We did the Hardy Weinberg lab where we had all the orange fish and white fish and spotted fish. That's this. So here we go. Imagine a wildflower population that has two different varieties of blooms that are different colors, right? So this is the same species, obviously, because it's the same population, and they come in two varieties, white or red. Imagine that in your head. Let's assume that the allele for red flowers is dominant to the allele for white flowers. The genotype frequencies can be calculated from the allele frequencies if the gene pool is stable / not evolving. And that is true, but I'm going to cross through this because I want to simplify things for how you study and how I want you to understand this. Yes, technically this is only accurate if there is no evolution, but just put that in the back of your mind. I just want you to understand how this works, and that's a little superfluous information. Basically, what we're saying here is we can calculate the genotype frequencies, right? We can calculate the percentage of in that population, the percentage that are homozygous dominant, right? Or the percentage that are heterozygous, or the percentage that are homozygous recessive, right? Because those are the three different types. Those are the three different genotypes. We can calculate that, and that's what we did in the lab, and that is where we come to the Hardy-Weinberg formula, right? And I already briefly talked about this in lab, and here it is again. This is the Hardy-Weinberg formula. You need to understand how it works and what it's used for and what everything represents, but I'm going to ask you I'm probably not going to ask you any mathematical questions about this, but if I do, it's going to be really easy. Even though I'm going to show you this on the next slide, I'm going to go ahead and tell you now. P squared represents homozygous dominant, and let's just use, I don't know, T. So that would be, all the people or all the organisms in the populations that are homozygous dominant, big T, big T. The middle one represents all the heterozygotes. So all the members of the populations that are big T, little t. And again, the T is just an example. It could be any letter. And then, of course, by process of elimination, this here, Q squared, that represents all the homozygous recessive. So in this example, little t, little t. And they all have to equal 1. or in other words, 100%. So really quickly, you might get some, this would be the easiest or the hardest the math would be. So I might give you a word problem and say, okay, in this population, 25% of them are homozygous, um, homozygous dominant. And I might say 25% of them are heterozygous. Now, what percent of them are homozygous recessive, right? And basically, it all just has to add up to 100%. So I'm not even take the time to ask you, but hopefully somebody would figure this out quickly. 50%, right? You don't even need a calculator, right? Because 25% plus 25% is 50%. So 50% plus what is 100%? 50%. And that's what the Hardy-Weinberg formula basically is. I'm not going to ask you any questions yet because the next slide is basically the continuation of this slide. There it is, right? That's basically what I was saying. Yeah. So another thing I probably won't ask you, even though it is on the study guides, is I'm not going to necessarily need you to remember that this one is the dominant, this is heterozygote, this one's the recessive. If anything, like I said, I might give you some word problem where I just say the percentage of dominance is this, the percentage of heterozygotes is this. So what is the percentage of the recessive homozygotes, right? Anyway, any questions about that? Any questions about the Hardy-Weinberg? All right, another quick example, and I want to talk about really quickly, I'll say this. So here again, we're talking about genotype frequencies. If we're talking about allele frequencies, it's even easier, right? So you're either the dominant allele or the recessive allele, and it all has to equal to 1. So again, if I give you a math question, it'll be really easy and even easier than the genotype frequencies, because if I say 75% of this population have a dominant allele, then what percent has the recessive allele, right? So what percent has a recessive allele? Hopefully that should jump out at you. 75 plus 25 equals 100. Any questions about this? Okay. Now let's talk about why that's important, because obviously for the exam, it's not that important. And because none of you are biology majors and you probably never use that again, it's not that important. So really quickly, I'm going to explain why it's important. I'm probably not going to ask any questions about it on the exam. But the Hardy-Weinberg formula is used to calculate the percentage of humans with alleles for certain inherited diseases. So again, for you guys, that doesn't directly matter because you're not doing these calculations, but this does affect you for as far as public health is concerned, because this is how people who are involved in public health can tell us with certainty, like, X percent of the population has this allele. And this is how they can figure it out. And obviously that would be important. And I wish we had more time to talk about it, but you can look that up for independent work. Why is it important to know what percentage of people have a certain allele for a gene? Anyway, the example that your book gives you, and put an X to this really quickly, just as a visual reminder that I'm not going to ask you anything about PKU. But PKU is a disease that gives you with the inability to break down amino acid phenylalanine. So, you need to be able to break it down for various reasons. Let's just, for now, just say it's bad if you can't break it down. This disease is caused by a recessive allele, right? So that means, let's call it, I don't know, R. That means you would need 2 little r's to have it, right? Big R, you wouldn't have it. Big R, you wouldn't have it. You would need 2 recessive alleles to have it. What we know, by the number of people who are born, 1 out of every 10,000 babies in the U.S. has this disease. So then we can use a little bit of algebra to figure some stuff out. So again, sorry, this looks like garbage. P squared plus 2PQ equals Q squared equals 1, right? So we know, again, Q squared, that's the recessive, right? So we know 1 out of every 10,000 babies is this, right? Little r, Q squared. So that means Q squared equals 0.0001, right? Because 1 out of every 10,000, that's what that equals, this number right here. So again, we're just doing algebra. We want to get rid of the squared, so we take the square root of both sides. The square root of Q squared is Q. The square root of 0.0001 is 0.01. And now we want to, again, I gave you the wrong one. I did all that and it didn't even matter. No, that'll work. Sorry. Anyway, so we take away the one. We take one away from that. That leaves us with a p, where m1 minus q equals p. We already know, therefore, that p has to equal 0.99. And because we know that, then we can fill in the blanks and figure out that 2pq equals 0.0198, which is about 2% of the population. And I went through that quickly, and I know that's probably slightly confusing with you or for you, but that's okay, because my point isn't to show you how the math works. My point is just to show you, hey, if you use algebra, we can figure out things about population health by using the Hardy-Weinberg formula. So long story short, even though you'll probably never use the Hardy-Weinberg formula, it is important for public health. The end. And let's see the next. No. So any questions about this slide? There shouldn't be, because again, I'm not going to ask you anything about it. All right, let's talk about microevolution as change in a gene pool. And again, we've already done this when we did the fish lab, right? The orange fish versus the white fish versus the white or the orange spot fish. A non-evolving population is what it's in genetic equilibrium, which is also known as the Hardy-Weinberg equilibrium. I might ask you that, I might not. I did bring that up in the lab. That was part of the introduction, right? So again, when something is not evolving, it's said to be in the Hardy-Weinberg equilibrium. The reason I'm probably not going to ask you anything about it is because this is a 100 level non-major biology class, so you don't need to know too much about Hardy-Weinberg, because it's not going to be that important in your life. I also point out too that this is all like theoretical. There is no population that's ever in equilibrium, and you'll see why when I tell you the conditions that it has to meet to be at Hardy-Weinberg equilibrium. So again, this is when the gene pool remains constant from generation to generation. So even just talking about, even if we're, the gene pool talks about all the alleles, right? All the genes and all the alleles. But even if you would just consider the one gene, right? And the example I've used before was chin dimples, right? Even among just that one gene, imagine the human population over time. Even just that one gene, the percentage of people who have chin dimples versus the people who don't have it, that's going to change, right? And that's just one gene. And then think about, we have tens of thousands of genes. So obviously that's not going to be happening. And another way of saying this bullet point is this, right? These two bullet points are basically saying the same thing in different ways. The frequency of alleles, P and Q, right? So dominant versus recessive. That looks more like a P, but whatever. Dominant versus recessive. And the genotypes. So again, homozygous dominant, heterozygous, homozygous recessive remains unchanged, right? So we know that's, again, like I said, this whole conversation is theoretical. It's academic. It doesn't really happen in real life. But because generation to generation change and the allele frequencies is evolution on the smallest scale, we refer to this as microevolution. So that lab we did was called basically that was about microevolution, right? We weren't talking about those fish turning into new species. They were the same species. It's just that the gene pool changed, right? The percentages of orange fishes versus white fishes versus spotted fish. But any questions about this slide? Okay, let's talk about the mechanisms of evolution. And again, we've already discussed this in that lab. So this is a quick review. And in my opinion, it's pretty simple. These are pretty simple concepts. What mechanisms can change a gene pool? So obviously we know about natural selection. We've talked about it, talked about it, right? And again, natural selection, as far as the exam is concerned, is probably the most important. And it's also important in real life because it's the only one that promotes adaptation, right? So all this other stuff is not necessarily like the best gene or the best allele is chosen. It's different, and you'll see how as we talk about it. And the first one we're going to talk about is genetic drift. You need to know the difference. And before I jump into this, you need to know the difference between natural selection, genetic drift, gene flow, and natural selection. Well, I already said natural selection, but yes, you need to know the differences between these things, because these are all ways that populations evolve, and you need to know the difference. So genetic drift, that is the change in the gene pool. I'm missing a word there. Gene pool due to chance. Meaning, again, like before I share that really quickly, using those orange fish, that was the first experiment we did. There was no advantage to being orange. There was no advantage to being white. There was no advantage to being spotted. And we just wanted to see how the numbers changed over time. And they did change a little bit, right? which was all by chance because there was no advantage or disadvantages to being a certain color. But here we go. Here's your book example. Um, and your next word for attendance is flower. You can see a bunch of them. We're looking at them here. So here we go. Here we have that population again. We have red flowers. We have white flowers. They're the same species. Red is dominant to white. So here we can see how many flowers we have. We can see what color they are, right? So we can see their phenotypes because, phenotypes is red or white. We can see their genotypes, right? Heterozygous, homozygous dominant, homozygous recessive. We can see them all. And there's no advantage to being red or white. There's no disadvantage to being red or white. It just so happens that by chance, these ones that are squared off, those are the ones that just happen to breed to make the next generation, right? There was no advantage to the colors. So then we can see that the generation one, the change, there was a change, right? We went from 70-30 to 50-50, all by chance, because again, there's no advantage or disadvantages to being the colors. And then by chance, these two flowers happen to be the only ones that mate in that generation. So then we go to the third generation, and there we go. Now we really have a bit change. We went from 70-30 to 50-50 to now 100% of the flowers are red and 0% are white, all by chance. There's no advantage. So Cliff Notes again, genetic drift is a change in the gene pool by chance. That's what you need to know for the exam. So any questions about what genetic drift is? All right, pretty simple concept. So let me make it a little bit more complicated, right? We don't want the exam to be too easy. So here we go. So let me back up. There's two different types of genetic drift. Yes, genetic drift is a change in the gene pool by chance. And yes, you do need to know that, but you also need to know that there's two different types of genetic drift. There's the bottleneck effect and the founder effect. And you need to know, first of all, that they both fall under genetic drift, and you need to know the differences between them. And they're similar in a way, but they're slightly different. And you need to know the differences. So here we go. First one is the bottleneck effect. Again, that's an example of genetic drift. We already said that. The bottleneck effect, and this is the part that you need to know for the exam, results from a drastic reduction in population size. That's hence the name bottleneck, because the bottleneck kind of passes through, or the population passes through a bottleneck, right? So that's the severe reduction in population size. So imagine I'll show you a picture of an actual bottle. So in a different term, imagine like a 5-lane highway being narrowed down to a 1-lane highway, right? So you have a lot fewer cars coming through now because of that. But anyway, I'll just leave the examples to the book once we get there. So again, for the exam, this is the most important thing. Everything else I'm teaching you, it's good to know, but I'm probably not necessarily go into this much detail. But why is it important, right? So when you go through a bottleneck, this decreases the genetic variability because some alleles are lost from the gene pool, and that's not good, right? We want genetic variability. I could go on and on about that. We don't have time for it, but basically, genetic variability is great because I'll just use COVID as a quick example. Some people got COVID and died. Some people got COVID and were asymptomatic. Some people got COVID and they got a little sick, right? And there's a lot of other reasons, but that's also due to a lot of the genetic variability between humans. Some people are just more susceptible to that disease. And it's the same with any population. So having that variability might help a population survive a certain disease, amongst many other things. That's just one example. So the results in the loss of an individual of individual variation, again, hence adaptability. So again, had humans gone through a bottleneck, maybe World War II, let's say World War II turned out worse than it did, which says a lot because it was pretty bad. Let's say we lost even more of the human population, and then randomly the people who had the genes in them that would help them survive COVID, even though COVID didn't exist back then, let's say randomly they died, way back in the 40s. And then those alleles are just gone. So then cut to 2019, we have this new disease, and then everybody's dying from it because randomly, back in the 40s, we lost the allele to have us be able to resist or the adaptability, right? The ability to resist that disease. So we're going to talk about this a little bit more on the next slide. I'm going to give you some visual examples, but do you have any questions so far on this slide? Again, for the exam, I'm giving you a lot of information, but for the exam, the bottleneck effect is one of the two types of genetic drift that we talk about, and it results from a drastic reduction in population size. So here we go. This is a good visual example. Here we have the normal population. We've got a green allele, we've got purple alleles, we've got orange alleles. You can see them right there. You dump it out, um, and there's no advantage to being either one of those colors. So just randomly we see what comes out. And it just so happens that we lost most of the green alleles, we lost all the orange alleles, and now we're left with mostly purple alleles and one green allele. Just by chance, right? There was no advantage to being purple. There was no disadvantage to being green or orange. That's just what happened to be. And that is a bottleneck. Your book gives you a real life example other than um, bottles, right? But I'm going to go ahead. So I'm not going to ask you anything about this. It is just an example, and there might be questions about it on the study guide, but I don't think there will be there won't be on the exam. But anyway, real life example of this is cheetahs, because they've had at least two genetic bottlenecks. The last one, or excuse me, one of them happened at the last ice age about 10,000 years ago. Obviously, it was just a little bit too cold for them, so most of them died off. And then in the 1800s, the farmers hunted them nearly to extinction. And especially, this is an even better version, I think. I won't get into why, but you know, if they're just getting killed off randomly by hunters, again, there's no advantage to being this or that. They're just getting killed by the hunters, and you only left a few of them. Then that means they lost a lot of their genetic variability. So because of that, they've reduced their capacity to adapt to environmental challenges. Because again, you need variation to be able to do that. Sorry, any questions about that example that I'm not going to be testing you on? Okay. Let's talk about the other version of genetic drift, which again is a change in the gene pool by chance, right? There's no advantage of this or disadvantage of that or sexual selection or anything like that. Let's just talk about genetic drift, the version called founder effect. And it's in the name. This one's pretty easy. The founder effect is when a few individuals colonize an isolated habitat. Let me just focus it down to you like this. If you take notes and like to do flashcards, you can do founder effect when few individuals colonize. You just even leave it at that, right? Or if you're going to write it in words that maybe make more sense, um, when few individuals start a new population. You can even think about it as that, because I don't like this idea of colonize, because it's not necessarily always colonization. And the isolated habitat part can be tricky, as we'll talk about later. So just think about when it's founder effect, just think about what a founder is, right? Think about like the first Native Americans. Of course, they weren't Native Americans at the time, but the first Native Americans that crossed over the land bridge from Asia into North America, right? They were the first humans in the Americas, right? So they founded, so to speak, humans on this continent. Anyway, here I am talking and talking, but right here, this is another great bullet point to briefly define founder effect. It represents genetic drift in a new colony. That's good. And I'm not going to ask you these, but these are great examples of some genetic drift. It explains the relatively high frequencies of some inherited disorders in small human populations. And I like this because we're talking about humans, and I love that because we are humans, so that we relate to it. But keep in mind that the founder effect it can happen to any organism, not just humans, but we're using humans as examples. And if you were to download this PowerPoint, these are links. You can click any one of them and watch them. So there's an island in the Pacific called Pingelap, and the people who just so happen to, like, the first humans to get there, just so happen to have a higher percentage of colorblind people, right? So because of that, these people to this day still have a higher percentage of colorblind people than the rest of the human population. The Amish, they have a few things. By the way, these are all great independent work topics if you want to look into them. Amongst other things, there's a higher percentage of polydactyly among Amish, and that means people with extra fingers, right? So, I've got 10 fingers, 5 on each hand. Some people have extra. And the percentage of people who have extra, let's just call it, and I'm making this number up, but just to say 1% of the population of the world has extra fingers. But in the Amish community, let's say, I don't know, 5%. I don't know what the actual numbers is, but the point is they're higher because the people who founded the Amish community happen to have a higher percentage of it when they founded it. In Martha's Vineyard, the percentage of people who were deaf that founded that colony happen to be a little bit higher, a higher percentage. And still to this day, there's happens to be a higher percentage of deaf people there than in the general population. And that's actually where American Sign Language was invented. So, you know, again, for the exam, the important thing here is the founder effect is the genetic drift in a new colony. I don't know. To me, this is really interesting stuff. If I was going to write independent work, I would try to look into these examples and look into other examples. What are some other founder effect examples? And not just for humans, but other organisms as well. Any questions about this slide? All right. Next thing we're talking about is gene flow. This one's also, in my opinion, pretty easy academically because it's in the name. Genes are flowing, right? So gene flow, again, it's just another source of evolutionary change. So it's not natural selection. It's not genetic drift. This is gene flow. It's its own thing. What is it? Gene flow is the genetic exchange with another population. So sometimes it could result, and that's the most important part, right? As far as the exam is concerned, that's your bullet point right there. Genetic exchange with another population. Let's see. The next word for attendance is right here. I'm circling it. That's the next word for attendance. So anyway, yes, gene flow is a genetic exchange with another population. That's what you need to know for the exam. I'm going to teach you a little bit more about it. Like I said already, it may result in the gain or loss of alleles. Obviously, if you have genes flowing into your population, you're probably going to gain some alleles. If you have individuals leaving your population, you're probably going to lose some alleles. And it also reduces the genetic differences between two populations. So imagine there's some island here and an island here, and there was a cruise one day. There's a cruise ship. Actually, this part happened. There was a cruise ship with a bunch of NBA stars, and if you remember, I think I'm remembering this correctly, back when COVID-19 first happened, they were isolating themselves, and I do think some of them lived on a cruise ship at one point for some reason. Anyway, let's just say there's a cruise ship with a bunch of NBA stars. Obviously, the percentage of tall people on that cruise ship is going to be higher than the percentage of tall people for the regular population. Let's say it crashes and all those people go live on this island and they're never saved. So for hundreds of years, we have a population of really tall people. Meanwhile, there's another cruise ship around the same time. We've got a bunch of horse jockeys, right? A bunch of short people. And they also crash, and they all live on this island for hundreds of years. So here we go. We've got a population of really tall people, population of really short people. They very rarely, or they don't ever communicate. And then one day, one of them gets brave enough to make a little raft and go exploring, and they come here and like, oh my goodness, look at all these short people. And they start going back and forth. And because of that, instead of having really tall people and really short people, once they start interbreeding, that reduces the genetic differences, and maybe these taller people start getting shorter. These shorter people start getting taller. Just an example. If you download the PowerPoint, you can click this where it says another short video on evolution, including gene flow. And I recommend it. They're really short, really easy to watch. But any questions about this slide? All right, go figure. We're going back to talking about natural selection. And again, I teach out of the book. This is the way your book teaches it. So we do talk a lot about natural selection because it is very important. Because it is only natural selection that leads to adaptive evolution, which is evolution that results in a better fit between organisms and their environment. So you might say natural selection is the only thing that makes populations better, if you will, right? Everything else is just random. Founder effect, gene flow, that's just random, right? There's no advantages to that. Your book gives this example. I'm going to put it next to it because I'm not going to ask you any questions about it. You can look into it more if you want for independent work. But the example your book gives about natural selection is the blue-footed booby. It's an excellent example of adaptations for its home. Why? Because its body and its bill is streamed like a torpedo, which is important because the way this thing eats is it kind of flies above the water. And then once it sees the fish that it wants, it kind of does a nosedive into the water and it clicks it out, right? So it would be important to be streamlined like a torpedo. So you're quick in the air, quick in the water. You can get deep, right? It minimizes the friction as it dives, so it gets you deep enough to get the fish you want. Also, if you get too deep and you're a fish, or excuse me, you're a bird and you can't breathe underwater, you're going to want to be able to stop when needed, right? So it has a large tail that can stop it. Like, oh, I got my fish. Let me stop and go back up to the air where I can breathe. So those are great examples of natural selection because those things, they have adapted to be good at their environment. Now, why your book started talking about this right now, I don't know, because we're talking about natural selection, but females prefer males that have the brightest blue feet. So if they have dull blue feet, the females aren't really attracted to them. And again, honestly, I don't know why your book points that out, because we're going to talk about this later. This is an actual example of sexual selection, not natural selection. But anyway, but then if you download this PowerPoint, you can click this video or click this link and watch another little short video about natural selection. Here's a picture of a blue-footed booby. See, you can see how it's nice and streamlined. It's ready to fly quickly and dive quickly and get through the water. Big tail that can serve as a brake, and it's bright blue feet. So any questions about the blue-footed booby? Even though I'm not going to ask you any questions about it. All right, so let's talk about what evolutionary fitness is. Specifically, the term relative fitness. This is the contribution individuals make to the gene pool relative to the contributions of other individuals, sometimes slightly inaccurately referred to as survival of the fittest. The reason it's inaccurate, let's think about it. If we're just talking about this bullet point, well, I don't have time to ask you, and you guys are being really quiet anyway. But if I were to ask you, what do you think this means? The contribution an individual makes to the gene pool relative to the of the other individuals. Excuse me, let me back out, see what that noise was. That's somebody from another class coming in early. All right, so I'll deal with that later. Sorry for the interruption, guys. Let's get back in here. Actually, while I'm at it, make sure I'm still recording. Yep, still recording. Hallelujah. Okay. Anyway, animation, so current side. If I were to ask you what this bullet point means, hopefully, basically, when you think about it, it's like, who's having more kids, right? Basically, that's it. Like, who's making more babies is what this bullet point means. Which, again, this is why I don't necessarily like survival of the fittest, because survival of the fittest is only half the story. If you're surviving, but you're not procreating, then you're not really contributing to the gene pool. So it doesn't matter what awesome genes you have that help you survive. If you're not reproducing, then it doesn't matter, right? So that's what relative fitness is that first main bullet point. So any questions about that first main bullet point? Again, you can just think of it as relative fitness. Who's having more babies, basically. All right, here's a whole different concept. It's a very simple concept, I think, but you need to know these three general outcomes of natural selection. Natural selection ends in three ways, basically. No matter what we talk about, basically it happens in three ways. So let's talk about it. In your book, this is just an example I'm giving you, but if we graph the coat color of a population of mice, we would get a bell-shaped curve. And I'll show you what I mean on the next slide. And keep in mind, this whole thing about the mice, it's just an example. This is the way these three things I'm teaching you happens with every example of natural selection. This so happens the example I'm giving you is mice and their fur color. So if natural selection favors certain colors over others, the population will change over generations, and there are only three general outcomes. And you need to know these. And I'm going to go through this slide quickly because it's going to be easier for me to visualize it for you and show you pictures. And I'll explain them more when I show you. But the first one is directional selection, which is when the population shifts by selecting in favor of an extreme phenotype, right? So if we were talking about humans, they're becoming taller, or humans become shorter. That would be directional selection. Disruptive selection, it would lead to a balance between two or more contrasting phenotypes. So in that case, again, instead of having like a bunch of like the way the human population is now, where we have a lot of medium-sized people with fewer taller people and fewer shorter people, disruptive selection would mean we'd have an increase in taller people, an increase in shorter people, and a decrease in those middle people. And then we have stabilizing selection, which is basically the opposite of disruptive selection. And stabilizing selection, we're actually favoring the intermediate phenotype. So again, these are all fancy words, but what I mean there, again, using the same example, we would have fewer tall babies, or fewer tall humans, fewer short humans, and even more medium-sized humans. That's what stabilizing would be. And that's what you need to know. And I'm going through this quickly because I'm going to show you a picture and describe it a little bit more when I show you the picture. If we're talking about the mice, right, here's the original populations. There's very few light-colored ones, very few dark-colored ones, and most of the population are medium colored. Now, let's say, I don't know, these mice live on an island, and it's a volcanic island, so there's a new eruption. So all the stones that used to be gray and stuff like that, they get covered with lava, right? And then that hardens and becomes dark. So now there's an advantage to being dark, maybe, right? So then because of that, the light ones are getting eaten more by predators, so the population shifts this way, right? We're going towards one of the extreme phenotypes. Now, mind you, the artist could have drawn this the other way. Maybe a blizzard hits, like there's a new ice age, so now it's advantageous to be light colored. So then there'd be fewer dark ones, right? That could have gone either way. Your artist just happened to draw it going towards this phenotype. So it could have been that way, but they just happened to draw it this way. That is directional selection. Disruptive selection, again, we're getting fewer of that middle phenotype, which is the gray one, then we're getting more of the extreme phenotype. So more dark ones, more light ones. The opposite of disruptive would be stabilizing. So again, we're getting fewer light ones, fewer dark ones, and more of the medium ones. Pretty simple, right? It's all in the name. Directional, you're moving one direction or the other. Disruptive, you're disrupting the way things are. Stabilizing, you're stabilizing the way things already are. So any questions about this slide? All right, last bullet point. Let's talk about it. Sexual selection. I hope that I can finish it in a minute. You can leave at 8:50 if you want, but I'm going to go ahead and finish this since we're recording. Sexual selection. Well, first of all, the next, or I guess last word for, possibly last word. Yeah, last word for attendance will be traits. Sexual selection is defined as a situation in which individuals with certain traits are more likely than others to obtain mates. So again, it's not necessarily that it helps you survive, but for some reason it helps you get mates. Which brings us to this next term, sexual dimorphism. That is a distinction in the appearance between males and females. Like think about birds, right? Usually the male birds are brightly colored and pretty, and the female birds are pretty, or not, excuse me, not pretty. Female birds are dull. And again, it's not necessarily associated with reproduction or survival. Otherwise, that would be natural selection. If you download the PowerPoint, you can click that link for watch another video about evolution. For some species, there's a secondary sex structures that are used to compete with members of the same sex. It's usually the males. And if we had more time to talk about it, I would explain why. You can look it up for independent work. Why is it usually males that are competing and females that are not? It should be pretty common sense. Anyway, we're talking about, here's an example of peacocks, right? They have ritualized display. The bigger the tail, the more likely they are to get laid, basically, right? And that is a good example of sexual selection, because other than that, there's no advantage to having a big tail like that. Those things live in India, right? So, or they're native to India. So that's a good way to get seen by a tiger and also make it hard to get away from a tiger. And of course, there's also combat, and I'll talk about that on another slide. More commonly, individuals of one sex, again, usually the females, are choosy in selecting the mates. And again, if you were to download the PowerPoint and click that peacock, there's a video that talks about sexual selection. Here's the sexual dimorphism. See, right? Here's the male, it's brightly colored. Here's the female, it's dull, because it's the female that's doing the choosing. So it's the male who has to be impressive. And then again, like I said, combat, right? That's what I mean by combat. I don't mean literally animals out there with guns, but you hear these things are headbutting, and the winner gets the mates. And that's it. So any questions about not just this chapter, but anything in general? This is a weird time for our semester, for our course. So if there are any questions while we're recording, now's the time to ask them. Okay. So again, this week's labs, it's already been posted. It's online. It's really easy. You're just watching a video. Yeah, Cedric, I saw that you raised your hand. What's the question?
It was just to ask, you said that we were going to be doing a review for the final quiz, correct?
Yes. Well, yeah, just like we've been doing. Yeah.
Awesome.
Most likely.
I just want to make sure.
Perfect. Great question. Yeah. So we will do, or most likely to be like it has been, where I'll post a video of an exam review, but that's just the exam review. Any individual one of you can come to me and meet me online so we can go over your old exams. And again, you'll get not only get the correct answers for the old exams that you can study, but then you can get that 5% boost to the grade. Any other questions? All right. So again, lab is online this week. I don't know what we're going to do next week. It's another virtual lab next week. So in theory, you don't have to come in, but I'm not making that decision yet. We'll see what happens. But I should be posting that soon today. So any other questions? All right. I will see you online on Friday morning. We will not be meeting in person. I still technically have to be isolated because of COVID.